This is part 6 and the final uh, section of the chapter on cells. I guess it's chapter 3. And let's go back. Okay, and the reason, by the way, I try not to make my lecture so long, but when I teach this information in biology, it covers at least five chapters that I can think of, and I'm having to cover all of that information, give you a good review of all of that information for the TEAS test in one chapter. So if sometimes I seem like I'm talking a little fast, you can always stop, and go back. I mean, at least this is recorded, so you can always go back and listen to something again, or anything that you need to do. But anyway, I better get started or I won't get this one done in 15 minutes. What we have here are the stages of a cell's life cycle. Now, as you can see, if you look at it by percentage, about 75% of the life cycle of a cell is spent in a stage called interphase. And when the cell is about to divide, it's going to be in interphase for up to like a day maybe or less when it's about to divide. But any other time it can go, if it's not going to divide again, it can go into a stage called G0, which means it's not going to divide anymore. But for a cell that's going to divide again, it will go into interphase and it'll spend about 8 or more hours in G1. G1 stage means that the cell grows and duplicates its organelles, because if it's got to have 2 copies of DNA, it's also got to have 2 copies of everything inside of it. So G1 is a growth phase. The S is the synthesis phase. DNA is synthesized or replicated, and that can take 6 to 8 hours. And then protein synthesis occurs in G2. Specifically, these proteins are proteins that, like the microtubules that form the spindle fibers. This is when the proteins that are needed for cell division are produced, and that takes 2 to 5 hours. And then you have mitosis and cytokinesis, which occur anywhere from 1 to 3 hours. And this is for cells that are actively growing, and all cells have different growth rates, but this would be like, for example, the first cell that resulted in you was called a zygote. It came from your mother's egg and your father's sperm, and that cell divided and divided and divided into the trillions of cells that you are now. And that's what I mean. All these cells have to divide in order to produce the final mature organism. So every time they divide, they go through interphase, and they go through G1, S, and G2 of interphase, and then mitosis, prophase, metaphase, anaphase, and telophase, and cytokinesis, which is the actual splitting in half of the cell. Mitosis is defined as the nuclear division of somatic or body cells. This would be any cells besides the cells that produce sperm and egg. Stem cells are cells that divide rapidly. Now, there are embryonic stem cells like the zygote. A zygote is an embryonic stem cell, and it divides into a ball of cells called a blastula and then a gastrula. And all of these cells are stem cells. They're able to divide rapidly and produce more cells. We also have stem cells that only divide and produce skin, or stem cells that only divide and produce blood. So some of them are differentiated. In other words, they'll only divide and produce certain types of cells. Some cells are programmed to self-destruct. For example, when the embryo of a human is at a very, very early stage, it has webbing between its fingers and toes. And so the cells that form the skin of that webbing are genetically programmed at a certain point to commit suicide. They genetically are programmed to commit apoptosis or cell suicide. Now, meiosis is cell division of sex cells, the cells that result in sperm and egg or ova. The first stage of the cell cycle is called interphase. It is the time between cell divisions. The cell spends the majority of its life here, 75% compared to how much it spends in mitosis. G1 is the growth phase, and it's also when the organelles are duplicated. S is when the DNA is replicated. Stands for synthesis. G stands for growth. And G2 is when the centrioles are replicated and the proteins that are needed for cell division are synthesized, like the microtubules or spindle fibers. DNA replication is when a strand of DNA completely copies itself. So what you have is the strand DNA, of course, is double-stranded, so that double strand has to be separated and it has to unwind. The enzyme DNA polymerase will bind to each old strand of DNA and add nucleotides to produce a new strand. And so you end up with, if this was your double-stranded original DNA, you end up with 2 sets of double-stranded DNA, and each one of those sets contains one old strand of DNA that was used to make the copy and one new strand of DNA that was created by DNA polymerase. Mitosis is the division of the chromosomes after DNA has replicated. So they divide so that each new cell will get a copy of the same amount of chromosomes that the parent cell contained. This occurs in 4 stages: prophase, metaphase, anaphase, telophase, and then cytokinesis is division of the cytoplasm. Mitosis is division of the nucleus. Cytokinesis is division of the cytoplasm. What happens in prophase is that the DNA, which during interphase was called chromatin, condenses into chromosomes. So chromosomes form. And when it says here they become visible, that's because they are coiling tighter and they are packing together into a structure called a chromosome rather than chromatin, which is very spread out. Each of 2 copies of DNA is called a chromatid. Chromatids are connected at the centromere. Another thing that happens in prophase is the nucleoli and the nuclear envelope disappear. The centrioles begin organizing the microtubules into spindle fibers. The centrioles move to opposite poles, and the chromatids attach to spindle fibers at their centromere. I was hoping there'd be a picture. Oh, there is. Okay. Then we have anaphase. The centromere of each chromatid pair splits. I'm so sorry. Then we have metaphase, and that is when the chromosomes move to the metaphase plate, which is the middle of the cell. So the paired chromatids line up along the plate. In anaphase, the chromatid pair splits and they separate from each other. Now they're called daughter chromosomes, and they are pulled apart and start moving toward opposite poles of the cell. Telophase is opposite of prophase. So instead of the nucleus disappearing, new nuclear membranes form, the nucleoli reappear, the DNA uncoils and forms back into chromatin, which is the uncoiled or uncondensed form of DNA. And the cell prepares to enter interphase again. Cytokinesis is the division that forms 2 daughter cells. And this shows you the stages, very small, but it shows you the stages of cells in interphase and prophase, metaphase, anaphase, and telophase. So in interphase, The DNA is in a form called chromatin. You can't see the individual chromosomes. In prophase, by the end of prophase, you've got centrioles that formed that are now at opposite ends of the cell, and they've all these little fibers that you see that are look kind of like a football shape. I just can't draw very well, but all these fibers in the middle here, they are the spindle fibers. They're organized and they connect to each chromosome, and those chromosomes are red and blue. They look like X's because they're attached at the centromere. So in this cell, there are 3 chromosomes. In metaphase, they line up at the middle. So in prophase, they become visible. Chromosomes become visible. The chromatin condenses and forms chromosomes. Metaphase, they line up in the middle. Anaphase, they're pulled apart. The sister chromatids are pulled apart. In telophase, they have arrived at opposite poles and new nuclei begin to form around the 2 sets of chromosomes. Now, each daughter cell, the parent cell had 3 chromosomes, and each daughter cell is also going to have 3 chromosomes. Cancer is mitosis, which has basically gone out of control. It begins with a tumor or a neoplasm, which is a mass produced by an abnormal cell growth and division, which is abnormal mitosis. If the tumor is benign, then it's not going to spread, and it's usually not life-threatening. It's rarely life-threatening. Now, it can be life-threatening if it's in the brain, for example, and it's taking up space in the brain and pushing against, causing pressure against a very important area of the brain. Some benign tumors can be life-threatening, but most are not. Malignant tumors are the ones that spread. So they spread from the original location, and that process is called metastasis. We say that the cancer has metastasized. And cancer is actually referring to just malignant tumors. When you use the term cancer, that automatically tells you that the tumor is malignant. Cells, in the beginning, when the zygote divides, the original zygote has not differentiated into a special type of cell yet. But after many, many divisions, all of those stem cells, embryonic stem cells, begin to specialize. That's called differentiation. They begin to become blood cells or skin cells or muscle cells. And when many cells that are similar to each other collect together in a group and sort of are coordinated in their action, like a collection of muscle cells or a collection of epithelial cells, which we'll learn about in the next chapter, can form tissues, can form muscle tissue or nervous tissue or connective tissue or epithelial tissue. Okay, so I'm true to my word. This is the end of chapter 3, or the cell structure and function.